Thanks.
Time as any, Mr. Newman. Take her down to periscope depth.
Yes, sir. Clear the bridge. Dive.
Stand right.
Greetings. Welcome to the Evidence and Reasons video channel. I'm Salvador Cordova, your host, and my co-host is Emory Moina.
Hello, everyone.
I'm sorry?
Just saying hello to everybody.
Oh, hello. Yes, Emory is a graduate student in biology, and like Emory, I'm a Christian, young Earth, young cosmos creationist, and we are running this segment, which I call Nerds Go Clubbing, after the term. There is a term in the American culture called clubbing that is going out to nightclubs. But among science researchers, there is a tradition of what they call journal clubs, where researchers will gather informally together and read through a paper together and comment on it so that they can stay up to date on the latest, or even sometimes revisiting historical matters that are relevant to the research. Why do we do this? Why do we do this? If I could summarize maybe one of the what does this have to do with the Christian faith? I thought about this today. I used to witness at James Madison University and also just hang out with the students. I would drive from Northern Virginia, Tyson's Corner, out to a campus that was about 2 hours away, just to talk to the students, share about faith, but mostly to listen. One reason I didn't talk much is I said I need to learn where people are. I need to hear what their concerns and hurts were, and it gave me a chance to think, well, how do I respond to this? One of the hardest things, it totally hit me out of the blue. A young lady at a free thinkers atheist agnostic group was just kind of sharing how she heard some insensitive comments from her peers. I don't even now remember the context of it. But she started crying. She said she'd been assaulted, and since this is a family-friendly channel. I'm not going to go into detail, but you know what I mean. Her eyes were just welling up with tears, and um, I and some of the other people present were really kind of speechless. We could only express our regrets and sympathy. And I was thinking, Lord, how do I tell someone like this about the love of God? This is the sort of person that I really want to reach out to. And to this day, and I'm sorry to say this, I've forgotten the young lady's name, but I think it was about 10 years ago. I pray for her a lot. Wherever she is, I was praying for her today. The Lord would give her, would be there to comfort her. So the question comes up, well, how could God allow this? And the New Testament teaches that this momentary light affliction is building for us an eternal weight of glory. This momentary light affliction is building for us an eternal weight of glory. 2 Corinthians 4:17. That for the believer in Christ, whatever terrible thing happens to a believer, it is part of the process of building an eternal weight of glory. We know that every great story, it's amazing when we think about great compelling stories, be they fictional or whatever, the ones that have been very captivating, if they have a happy ending at the end, it is more meaningful because of the tragedies along the way. Artists totally get that. And if God is the great playwright in the sky, this is part of the happy end. It may be hard medicine, but the pains in the early chapter are part of the happy ending. And what I'll say then is, for those who believe in Jesus Christ, God says how good it will be. There'll be no comparison with how good it will be to how bad it was. It'll be far beyond. So I know this is kind of hard to conceptualize, but just imagine how good heaven would have to be to make up for some of the bad things in a person's life. For the non-believers, there's not really much to say about how that will make sense for their life. That's even really the harder medicine, is that someone who doesn't know Christ, they go through bad things and then they face God's judgment on top of it on Judgment Day. But for the believer, God will work all things for the good, and he'll make sense of things. So my responsibility then, I mean, these might be nice things to assert, and the only way it will really count ultimately is if it's true. And part of studying creation science and archaeology and evidences and reasons is to show that the Bible can be trusted, that it was divinely inspired. Jesus said to study the works. If you do not believe me, believe the works. And I always found that's kind of funny. It's like, well, Lord, I don't see a lot of miracles. But now in the era of creation science, we are seeing the works of God. It also says that, yes, in Isaiah it says, surely you are a God who hides himself. And then there are passages where Jesus says, I thank you, Father of heaven and earth, that you've hidden these things from the wise and learned and revealed them to little babes. And I'm like, that's kind of a strange. I mean, it struck me. I said, why hide all this? It's ultimately to God's glory. But the way we learn about the invisible God is through the things that are made. And so that's why we do journal clubbing. Now, this is not necessarily the most pleasant experience. In some ways, it is bearing our cross to learn these things. And for graduate students, it really is bearing your cross sometimes. There are some classes that were pretty tedious. And so it's really great to have Emory here as we struggle through this together, as we read through it and say, hey, maybe parts of this are not fun, but in the end, you realize, oh my, we have learned something about what God has made. We learned something about his divine nature. And his invisible qualities. So that is kind of my goal for this journal club, not just to throw just data at you, to try to say, okay, look at it from this perspective. There are parts of it. I hope I've said enough to at least motivate you to take this in a different light. Because as you study this, especially the science students, I hope that it will comfort you that there is a God, and that he's all powerful and all wise. So let me share. I'm gonna. I'm in the midst of working on a paper, and I was looking up references. So combinatorial gene control. I'm not going to read the whole paper. And by the way, if you hear me doing mostly a monologue, it's because Emory here is still recovering from an illness, and I'm trying to save his voice. But I really appreciate him just kind of joining me and going through this. And I expect that he'll weigh in every now and then, but I told him I'm going to try to do most of the talking to spare his voice. God willing, when his voice is recovered, and I suspect he enjoys talking more than I do, I will happily give the floor to him. So what do evolutionary biologists tell us? And in the process of looking for material for a paper that I'm writing, I came across this paper called Combinatorial Control of Gene Expression. And it was in Biomed Research International. And you could tell by the names that this is from a different country. In fact, it says India, Department of Biological Sciences, Birla Institute of Technology and Sciences, Pelani, Rajasthan, India. So what do evolutionary biologists say? That life is badly designed? That's what evolutionary biologists say. But what do the actual researchers of the molecular and cell level have to say? So this was I'm just going to read the abstract. The complexity and diversity of eukaryotic organisms are a feat of nature's engineering. Well, they already got it wrong in the first sentence, but they got it mostly right. It's a feat of God's engineering. I like the word engineering, because engineering means design. The complexity and diversity of eukaryotic organisms are a feat of God's engineering. Pulling strings of such an intricate machinery requires an even more masterful and crafty approach. Masterful and crafty approach. And they go on to explain why. And I can't possibly read through the paper without getting bogged down in technical details. This is a non-trivial topic. But suffice to say, I keep seeing this pop up in scientific papers. And normally in scientific papers, you try not to use such descriptive words. You try to just be plain, boring facts. What this is telling me is that even the people studying this are so awestruck, they have to say something. It would seem irresponsible not to say something. So it says, only the number and type of responses that they generate exceed the staggering proportions of environmental signals perceived and processed by eukaryotes. Hence, at first glance, the cell's sparse stockpile of controlling factors does not seem remotely adequate to carry out this response. The question is how eukaryotes sense and respond to environmental cues has no single answer. It is an amalgamation, an interplay between several processes, pathways, and factors, a combinatorial control. A short description of some of the most important elements that operate this entire conglomerate is given in this paper. And so this is a short description, supposedly, but then it goes on and on. But remember the terms engineering, which means design, masterful and crafty. That is not what you'll get from Jerry Coyne. And he's embarrassed himself when he would be saying stuff about how something's poorly designed and it ends up being masterful and crafty. And I've often said that the way to understand biology is not, if we look at a Rube Goldberg machine, it's not designed for the benefit of the Rube Goldberg machine. It is designed for the amusement and for the glory, quote unquote, of the designer, not the designee. You don't build a Rube Goldberg machine for the sake of the Rube Goldberg machine. You build it for the benefit of the designer to show off his skill and his knowledge, his intelligence, his craftiness. And if we look at biology that way, it makes sense why it's put together. And so it is crafty, it is masterful. So I'm just gonna, okay, so let me, I'm just gonna blast through this because there's no way that we're gonna read through it right today. So he talks about all these transcription factors and these cis-regulatory modules, the post-translational modifications, chromatin modifications, co-regulators, about the 3D expression and control, long non-coding RNAs. Oh, they mentioned the PRC2. Yep, that's the polycomb repression complex 2. That's another marvel. So after all that, and this is just short, what do they say? The cell performs an amazing feat. Interpretation constantly varying external signals in keeping with an ever-changing pattern of gene expression. Amazing. All right, that's not the word you get from evolutionary biologists like Jerry Cohen that, or Dan Grauer, who want to call the genome junk. It always sort of amazes me that someone like Dan Grauer, it seems his purpose in life is to prove that his genome is junk. Just like, what's it? You know, what's up with you guys? That's what you want to do with your life. So let me just read the conclusion. The cell performs an amazing feat. Interpretation constantly varying external signals in keeping with an ever-changing pattern of gene expression. It does so with an intricate network of mediators and signaling molecules, those that are integrated together by using an equally complex system of signal transduction. The plethora of internal signaling required for gene expression is created by the cells by cleverly utilizing a comparatively limited reservoir of molecules. And I could go on, but just listen to those words. Masterful, crafty, engineering, amazing, complex, intricate, clever. The principle of combinatorial control of gene expression that is addressed in this paper sheds light on the mechanisms used by the biological systems to achieve tight regulation of the entire process. The cells use the limited amount of resources available at their disposal in the most innovative and economic ways to achieve the same. All right, it's just like, oh my goodness, is this like an ID promotion track? Seriously, I mean, I read papers like this all the time, and every now and then they just tell it like it is. I've been looking at I've been researching zinc fingers, and the word sophisticated and artful are used. I'm just like, that's kind of interesting. You know, there's not really a scientific definition of sophisticated, artful, or innovative, but it's hard to run away from this. So it says here, the cells use the limited amount of resources available at their disposal in the most innovative and economic ways to achieve the same. It points to a situation where the cells utilize a handful of molecules in various permutations and combinations to accomplish the startling task of constantly switching on genes and switching them off according to their needs. The controls of gene expression show complex and diverse patterns and are subject to various constraints such as factor availability. It is also noticed that although one tries to map the entire process by a linear series of events, the various steps are very close knit together. In such a scenario, it is virtually impossible to isolate a unique mechanism that governs the entire process. In the computer science world, we call this distributed control and processing. It's like the internet. The aim of functional genomics is to study the different mechanisms that are prevalent in some common biological systems and device models that encompass the different observations. And these models can then be used in various other fields of research application. This paper is an attempt to cover some of these mechanisms observed at the level of transcription initiation. The cis-regulatory modules, regulators, and co-regulators are at the foundations of these mechanisms. Emerging discoveries regarding the roles of link RNAs point to newer biochemical pathways and emphasize the notion of combinatorial control. These studies point to a new era, which might look to alter gene expression profiles by increasing the concentration of these RNA products within the cell. Each of these elements shows a variety and flexibility of function depending upon their conformational states. Additionally, the situation is made more interesting by availability of various regulators and co-regulators, which aids in the further selection of the combination required. The use of regulators and co-effectors with less than 100% binding efficiencies in systems whose performance is optimal for satisfying the cell's needs is a testament to the efficiency of the latter. The use of such complex signaling cascades also gives the cell greater control over the process as it now contains more than one regulatory step. Such a high degree of control is indispensable for the cell. The holistic understanding of these mechanisms will aid in improving the techniques used in rDNA technology, developmental biology, and so forth. Such improvement will aid in the development of applications that benefit humanity.
Can I hop in for a second, Sal?
Yes, by all means, sir.
All right. Folks, don't ever let anyone tell you that there is no evidence of design. Yes, I know the evolutionary biologists all run around out there making claims like, oh, there's no design, the human body's riddled with errors and all this other stuff. I mean, what's his name? Nathan Lentz wrote a book entitled Human Errors. The problem with his book is that, well, most of the errors he cites aren't errors. They're actually,
the errors are in his book.
Yeah, the errors are in his book. Yeah, they can go with that. But I mean, just reading through that paper, they can't, even though they really want to, although this may not be quite as strong because these folks are from India, so the anti-creationist bent may not be quite as strong in India as it is in the US, I don't know. But even though they want it to have evolved and they want it to there to be no design and they want there to be no God, they still can't avoid pointing to the design features because, I mean, did you read the end of that paragraph there? It's indispensable to the cell. Indispensable. In other words, the cell doesn't have it, the cell dies. Right? This is not, and what exactly the it is in that sentence is kind of immaterial. This high degree of control is in the cell. In other words, the cell has to have this. This isn't something that the cell can stumble onto and go, oh, this is a big help, I should use this. And you can go, and if you were to read through this article, you probably would find half a dozen things where it goes, oh, the cell actually really needs this. And if the cell doesn't have this, it kind of doesn't do too well or it dies. So welcome to design, where you can't just stumble onto things. You have to there has to be some forethought. And I know, Richard Dawkins, The Selfish Gene, I know all that. I've read them. Dawkins is desperately trying to cling to his atheism. And yet there is he does some useful science once in a while. Most of the time he's propagating atheism, but usually every once in a while he does some useful science. I don't know if he does anymore. I think he's retired now. I think the only thing he does is propagate atheism these days. But the point being, this paper right here clearly recognizes design, even though they don't appeal to it. They would never get published if they appealed to it. But if they don't appeal to it directly, but the language they're using, intuitively, they figured out that there is design here. And even if they don't consciously want to acknowledge it, the words they're using are design words. They're words that you don't use for a pile of chance random processes. Anyway, that's my little bit. But
And it's not just trivial design. I know why they're in awe. I've talked to biochemists. I said, could you build something like this from scratch? And they're like, no way. I mean, if we could, we would have cured all diseases by now. The machinery, the control, the intricacy is beyond our technology. We build computers, and in general has centralized control. We have a very hard time building systems that have distributed control. Internet's kind of like a model of distributed, but to have distributed control that is kind of, Bitcoin's another example of distributed control toward one single goal. But the way we see it, this distributed control, it's beyond anything that we know how to do. I briefly worked in passing when I worked for the think tank MITRE Corporation. MITRE stands for Massachusetts Institute of Technology, Research and Engineering. The joke is the RE actually stands for Rejected Engineers. So I worked for MITRE, MIT's Rejected Engineers in Tyson's Corner, McLean in Virginia. And I briefly worked in their nanosystems group. And they said the two outstanding problems they have in building nanomolecular machines that they would love to solve is to build self-healing machines and machines that can self-assemble. The cell eukaryotic organisms can do all that, and they can do it phenomenally. We don't know how to do that. We don't know. And so it doesn't surprise me that people will say, will describe this as an amazing feat, because biotechnologists and medical research want to know how to do some of this because they want to cure diseases. God bless them. But now they're confronted with the magnitude of the task because of the complexity of the cell. And so, yeah, this was a total accident. And I'm glad that, you know, even though I didn't get to all the details here, I want to give the readers, I said, okay, you know, I could have just been spewing out facts and you wouldn't have seen the forest through the trees. I'm glad I encountered a paper like this, because most of the papers we'll encounter, I'm going to read a section of the paper of more of what you'll get. Like I studied this hot air link RNA. Okay, I'm just going to read the paragraph, and this is what it will be like in a typical journal club. It'll say, the hot air link RNA is one example. The stretch of link RNA contains two sites, a 300 nucleotide stretch in its 5' end and a 700 nucleotide stretch towards its 3' end. The former is the binding site for the PRC2 proteins, while the latter forms the binding site for the LSD1 co-rest-rest complex. Both of these complexes catalyze histone tail modifications that repress gene transcription. By acting as a recruitment site for both of these complexes, the hot air molecule can mediate gene silencing by means of multiple mechanisms. Okay, so just imagine doing that for hours upon hours. But
sounds fun.
Oh goodness. Okay.
I'm sorry. I'm the nerd here. Everybody else, I'm sure, is going, what did you even just say?
Right. But you see what I'm trying to point out is look at it from a different way. When you get a parts list and you see how it's connected, you think to yourself, that is just amazing that all these parts can be organized to work in such an orchestrated manner and with such ingenuity. As an engineer, as I read this, when I was sitting in class, I wanted to jump up and say, praise God, praise God. I'm just like, yeah, that wouldn't be really appropriate in the classroom right now. But I was reading through this. I was looking at the hot air complex and I was seeing the diagrams the professor was showing. I went home every day after class and I was just thanking God. I said, you know, the world is a total mess, but you've left remnants. You've left artifacts that tell us that there is a God who's all wise, who's far smarter and more knowledgeable than all of humanity put together, because only a mind of that caliber would be able to do this. And we're told in the Bible that even that is perhaps a small thing for the Lord. He's not even showing the fraction of what he's capable of. So that's just kind of I'm trying to put a perspective here on why there's a journal club. And it's like, well, I'm trying to put a little bit more inspiration in this, because it can be very dry. A lot of the journal club is just reading through passages like what I just read about, like the hot air link RNA. But me being an engineer and putting things together, my jaw drops when I see the complexity, the intricacy, and just to use their words, the masterful, clever, crafty, innovative. Yeah, I see all that now. I see all of that now, and it humbles. So let me share something from Romans 1:20, and why, people backstage, Emory and I were talking about sometimes the resistance we get in church, because it's like, I wanted to teach Sunday school and declare the works of God in creation. And it's like, well, the attitude I get is like, well, why do you want to talk about molecular biology? Why don't you just talk out of the Bible? And that's sometimes what makes people uncomfortable with teaching of creation science and apologetics and evidence and reasons, because you actually talk about things that are not actually explicitly, they'll feel comfortable if you just read the Bible and just throw a few opinions. They'll feel comfortable with that. But to go into detail like what we did just in the last, say, half hour, that begins to kind of make some people feel uncomfortable. It's like that, a lot of them don't get it, and it feels irrelevant. But how do we learn? How did God design the universe to teach us about him? It says in Romans chapter 1, verse 20, for his invisible attributes, his eternal power and divine nature had been clearly perceived ever since the creation of the world in the things that have been made, so that they are without excuse. So if you want to learn something about God, his invisible attributes, his eternal power, divine nature, you can learn about them through the things that have been made. So this journal club is helping us understand the meaning of Romans 1:20. For his invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world in the things that have been made. And if I may, Matthew 6, I kind of like the Gospel of Luke. It's kind of my favorite. So Jesus said in Luke chapter 12, and he said to his disciples, therefore I tell you, do not be anxious about your life, which you will eat, nor about your body, which you will put on. For life is more than food, and the body more than clothing. Consider the ravens, they neither sow nor reap. They neither have storehouse nor barn, and yet God feeds them. How much more value are you than the birds? And which of you, being anxious, can add a single hour to his lifespan? If you then are not able to do a small thing as that, why are you anxious about the rest? Consider the lilies, how they grow. They neither toil nor spin. Yet I tell you, even Solomon in all his glory was not arrayed like one of these. Now, here we are in the 21st century. That verse has a lot more meaning now to the 21st century person. Okay, Jesus has a way of using words very economically. So just consider the lilies, how they grow. Consider the lilies, how they grow. All right, let's do that. You want to consider how the lilies, how they grow? Learn about combinatorial control of gene expression. that's Jesus' recipe for helping you so that you don't be anxious. You look at combinatorial gene expression, you will, if you have any sensitivity toward God, you study this, you will know that God is in control. Only a genius could put this together, and you'll be comforted. That's what it did for my life.
Amen to that. And just getting into the nitty gritty of molecular biology, genetics, fill in the blank of whatever field you'd like to get into when it comes to science. The deeper you get into the field, the more you start to pick up on potential aspects of design, potential aspects of a creation. And a lot of people just want to suppress the truth. They want to do what the Book of Romans says and just say, no, I don't want any part of that. I'd really rather not, because of for fill in the blank, whatever reason they have. What I found with atheists a lot of times is one of two things: either they know they're living a wicked, sinful life and they don't want to change, or they got hurt at some point in the past and they are angry at God. Now, not every atheist will fit into those two stereotypes, but that's a lot of them. And that's why a lot of evolutionary biologists don't want to admit to the possibility of design, because if they were to admit to the possibility of design, they would have to deal with the possibility of there being a designer, and then they might have to actually listen to what the Bible says, and that would be for a lot of them, that would be like the equivalent of a kiss of death. It would also destroy their careers if they're not retired, like somebody like Richard Dawkins. It would also destroy their careers. The only way they basically have to wait until they get tenure to do something like that, or either that or be John Sanford level genius. And even Sanford got a lot of pressure. Sal, I could see you want to say something, so go ahead and unmute yourself and say what you want to say.
Well, that gave me a break to kind of refuel here. I'm just going to try to save your voice. You alluded to the suppression. It says, going back, taking a little more of the context of the verse I quoted earlier, for the wrath of God is revealed from heaven against all ungodliness and unrighteousness of men, who by their unrighteousness suppress truth. For what can be known about God is plain to them, because God has shown it to them. For his invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world, in the things that have been made, so that they are without excuse. And we see that with like what happened with the whole ENCODE thing. They were starting to highlight the glory of the genome. Yes, there's been deterioration. So if they say there's junk there, I would actually absolutely agree. But no one knows the figure of how much junk. But we would suspect that more of it's functional than the evolutionists want to admit. And Dan Grauer, evolutionary biologist, just went on a tirade, and this $500 million project, the ENCODE project, it could be even more than that by now. He was saying that these are crooks and ignoramuses, and he just went on a tirade. And I was beside myself. I said, these guys are medical researchers. They're trying to solve cures to heritable diseases and cancers, and you're just calling them crooks and ignoramuses. Why? Because they highlighted what God had made. Mm-hmm. They didn't mean to, but they sure did. Go on, Emory.
Yeah, Grauer's motivation is very, very clear if you read the title of his paper, because it includes the statement as follows: the evolution-free gospel of ENCODE. Grauer is deliberately trying to smear these people because he knows if the genome is functional. He says this. He admits this. It's not like I'm making this up. He actually says if the genome is as much functional as ENCODE it says it is, then evolution's not true. And he wants evolution to be true so badly that he's willing to smear. And I had to use this man's textbook, by the way, in one of my grad classes. That was fun. Because, I mean, the entire first chapter of the book, which we didn't cover in the class, but was basically a smear on creationists. It was just the entire first chapter of the book. I was like, oh my goodness, this guy's vicious.
Wait, you had to study Grauer's textbook?
Oh yes. I still have it around somewhere.
Oh, please don't sell it. We need to keep it for.
Oh no, it's not going anywhere. I'm not selling any of my grad school textbooks. I'm hanging on to all of them. So
if I may ask, what's the title of the textbook?
Oh shoot, I don't remember.
Yeah, okay, this is funny. Professor said on an exam, he said, "This is extra credit. Tell us the name of the author of your textbook and the title."
That's a great
yeah, that's kind of
a great
embarrassing.
bonus question. I could have told you the author. I don't think I could have told you the title, but I can, if we want, we can actually review through that sometime and pull out some stuff.
Oh yeah.
Because I do have it, and it was a fun read, but it was also like, oh my, what on earth? And it was funny because my teacher in that class kind of didn't like Grauer that much. You could just kind of tell from the stuff she said that she thought Grauer was a little over the top. And I think she agreed with him on some stuff, but she thought his behavior was a little over the top. And I think like going after the NIH people who are working on the ENCODE project is just, it came out as a personal level, not as a scientific attack. Yeah. And he didn't attack their science. He attacked their intelligence. He called them creationists, even though I don't think most of them are. I don't think probably any of them are. And this, unfortunately, is what tends to happen in the professional science level when they can't argue. They'll just smear. And unfortunately, I mean, this is what you get with Grauer, and you get it other places too. But Grauer's "The Evolution-Free Gospel of ENCODE" paper is a prime example of that. He didn't bother to attack the ENCODE science. He just went straight forward and said, "Oh, they're abandoning evolution. We can't have that."
So, since we brought up ENCODE, one of the discoveries associated that led to ENCODE, ENCODE did not discover this, but it led to ENCODE, was the hot air link RNA. And in fact, one of the discoverers of the hot air link RNA, I knew his name. It escapes me now. I'm having a Joe Biden moment. I can't remember his name to save my life. Wren, I think his name is John Wren. He discovered the hot air link RNA, and he called it hot air because he thought, well, most of the genome must be junk, so this could all end up being hot air. I mean, it is an acronym for something now, but it was kind of a joke because he like a lot of researchers, he stumbled on things, and he found out it was functional. So let me just briefly point this out. And it's easy to lose this. It says the former is the binding site for PRC2 proteins. That's a polycomb repression 2 complex proteins. All right, so this stretch of link RNA contains 2 sites, a 300 nucleotide stretch in its 5' end and a 700 stretch towards 2' end. The former is the binding site for PRC2 proteins. Now, where is this all headed? It turns out hot air will connect to all these proteins, and they'll create this this complex called LSD1 co-rest-rest complex. But what ends up happening is there are all these sites on the DNA that don't code for proteins. They are just parking lots for molecular machines. So a lot of it's turning out now, we're realizing a lot of the functionality of DNA is to serve as well-addressed parking lots that they're reconfigurable for molecular machines. That's different than the old paradigms. And the complexity of this is just phenomenal. And we're only beginning to see this in detail, but we've suspected it. And the more we look, the more we find. And it was really kind of disturbing, Dan Grauer's response to just go after people that were just trying to save medical researchers trying to save people's lives. This is an example. I mean, I don't know how Grauer could live with himself. well, maybe, I can't read his mind, but it would really bother me to smear someone's career who's trying to save people's lives just because you don't like the scientific results of his work. These guys are hurting medical research, and this is what I'm finding, and I've been saying it. Evolutionary biologists are having a hard time justifying the relevance to science. This is a good example. That whole thing with Grauer, that's a good example. What relevance do you have to modern science and any benefit to society at this point? They have like zero. In fact, if anything, they showed themselves to actually be a hindrance to advancement of knowledge. So that's my little soapbox. So we have to try to say a few things like this to make Journal Club a little bit more exciting. Otherwise, you're going to get just hours and hours of just talking about molecules. So anyway, let me see if I could find, let me touch briefly on it, if that's okay, Emory. I'm going to change the subject to something from our last talk.
Sure, go ahead.
Let's see, in our last talk, and I know this sounds really amazing, I referenced this by Gary Felsenfeld. He is a very senior researcher at the National Institutes of Health. There are various forms of DNA, and one of them is called zDNA. It's a special kind of DNA. And guess who was working on the discovery of zDNA? Michael Behe. Michael Behe, as a postdoctoral fellow, was working under the supervision of Gary Felsenfeld in the research of zDNA. Felsenfeld had a very illustrious career, and as far as I know, he's still working. In 2016, some of his staff were my professors in the study of chromatin modification. I took a biochemistry class in chromatin modification, which we associate with the field of epigenetics. Now, the word epigenetics is just a total mess. Everyone has you use that term, everyone will have a different definition of it. It's not a stable word. And even Gary Felsenfeld's definition doesn't agree with what other people use, even though he's the senior researcher and he was a pioneer of the field of epigenetics. But I threw out an item that I know sounded kind of like almost too much to believe, and this is leading to perhaps a topic of another session. So let me show you the title of the article. Actually, this was part of our classroom study, was this paper. Since Gary Felsenfeld was the boss of my professors, I guess they had to sort of like, well, we need to have the students read what Gary Felsenfeld had to say about epigenetics. So this is the 2014 from Cold Spring Harbor, Perspectives in Biology, A Brief History of Epigenetics by Gary Felsenfeld. And in here, it's only one paragraph. The extent to which the nucleus and cytoplasm each contributed to the transmission of a differentiated state in the developing embryos was, of course, a matter of intense interest and debate. A self-stabilizing biochemical pathway would presumably have to be maintained through cell division. A second kind of epigenetic transmission was clearly shown in paramecia and other ciliates, in which the ciliary patterns may vary among individuals and are inherited clonally. Altering the cortical pattern by microsurgery results in transmission of a new pattern to succeeding generations. It has been argued that related mechanisms are at work in metazoans, in which the organization of cellular components is influenced by localized cytoplasmic determinants in a way that can be transmitted during cell division. I think you know where this is headed, Emory. This is a form of inheritance that is not directly under the control of DNA.
Yeah, this is almost Lamarckian.
Yes. Now, the reason this is really difficult for evolution is then they wanted to be neo-Darwinian in the sense it's like, oh, we could just model how things evolve just by talking about how the genes will mutate, no problem. They now have to account for how organelles and cell parts came to be, because if the cell part serves as a template for subsequent generations, one has to say, well, how did that cell part evolve? It's not just through DNA. We're beginning to touch on this, and I will touch on some experiments that have shown that inheritance there's some way that the structure, kind of like the way a 3D printer works, is, I mean, a 3D printer is like a paper airplane compared to the space shuttle, because the way the cell does this is just remarkable. We don't know. But just suffice to say, it will somehow, there's a machine, it's almost like a machine that can sense the way a 3D structure is built, and it's just going to go through there and try to copy it as is. Mm-hmm. So that's why you can take this ciliary pattern, change it surgically, and it's just going to copy that. We don't know how exactly it does that. So there are organelles in the eukaryotic organism, like the Golgi, the endoplasmic reticulum, the various membranes associated with this. It can copy that. And it leads to some really tough problems. And I only began to explore it. So there are all sorts of things that it's like, well, unless it exists, it's not going to duplicate. You can't, it now throws another level of difficulty for evolution, because like without a membrane bound organelle to start with, it's not going to evolve out of nowhere. And we could go through these membrane bound organelles. We've done some experiments with the Golgi apparatus, but anyway, I
find it very, very interesting that the paper that they're citing about talking about the paramecium is from 1965.
Yes.
We have known about this. We have known that some things might not be inherited or transmitted via DNA inheritance.
Yep, a long time.
1965. And yet the Darwinists never mention it, deal with it, want to talk about it.
Exactly. And it's not all their fault. I mean, people want simplistic answers, and it's like, oh, we'll just try to do this through all DNA. It's just easier. Because to think about it elsewhere, I mean, people kind of have a sense of, they have a sense of being reassured if they can comprehend something. And to add all this complexity, it's like, oh, that's very uncomfortable, because now you don't understand things, and you may not understand it in your lifetime. I mean, scientists take great pride in understanding how things work, and the way biology is constructed. It's making sure you're not going to understand, not in 1,000 years, not with all the technology of the entire planet. You're not going to understand it. It's that complicated. But yes, it's been suppressed. It's been suppressed. And maybe you and I have a chance. Maybe we should put this a little bit higher priority in our. I mean, would you be interested in talking about cortical? I have papers on it.
If you've got papers, Sal, I will be more than happy to look into this and to look it over and see what they have, because I think the aspect of epigenetic inheritance is something that really throws a monkey wrench into Darwinism. Now the other problem then, of course, becomes how long it's maintained, because I know with some epigenetic inheritances, it's only maintained for a couple of generations. So, but the whole concept is interesting, and I think at least some aspects, at least in this paramecium, it throws a nice little monkey wrench into the.
Well, if I could say this, first off, I try to not use the word epigenetics. It's so abused. In this case, I call it non-DNA inheritance, and there's several classes of it. Chromatin modifications, one of them, RNAs, but then this whole cortical pattern, they call it cortical inheritance. And that would be interesting to see how long this cortical inheritance could be inherited. That would be really interesting. if they could alter the ciliary pattern in a paramecium and do like a Lenski type experiment, 30,000, see if it's persistent.
Yeah, paramecium reproduce pretty quick. That'd be a fairly straightforward experiment to run.
Yeah. Oh my goodness. Well, if only the government were willing to finance such interesting studies. But you know what? God's helping us. I was talking with colleagues. I said, God's helped us along because the need for medical research is doing all the work that creationists would have wanted to have done. And not all of it, meant much of it. We've got a huge ally in medical research, the biotech industry, and the pharmaceutical industry.
Without them knowing it.
Without them knowing it, they have advanced the design argument beyond our wildest dreams. And so we may end up getting a lot of research on cortical inheritance, and we're going to see evolutionary biologists even be more irrelevant and angry, hopefully. So
I'm not sure how much more angry you can get than Dan Grauer's The Evolution-Free Gospel of ENCODE, but if you could do it, I'm going to be very interested in reading that paper.
Okay, so I think we're at about an hour now. However, if we include our backstage time, this is already about an hour and 40 minutes. How are you doing?
I'm getting to a point where I'm going to need to call it, Sal. I'm losing it here.
Oh man, just when I was getting warmed up. Okay, well, you know what that means? We can meet again and have more fun.
All right.
But what I wanted to close with then, and I want to thank you for joining me. There was this book by Richard Dawkins called A Devil's Chaplain. And it was taken from a saying, something that Darwin wrote. So this book by Richard Dawkins, a Sunday Times bestseller, it's called The Devil's Chaplain. And it says here in Wikipedia, the book's title is a reference to a quotation of Charles Darwin, made in reference to Darwin's lack of belief in how a perfect world was designed by God, and a reference to Reverend, quote unquote, Robert Taylor. So Robert Taylor is called the Devil's Chaplain. That was his nickname because he ended up becoming totally apostate and advocating that advocating the claim that Jesus Christ was the myth. He was among the first mythicists. Anyway, what Darwin wrote, referring to Robert Taylor, who is the Devil's Chaplain, Darwin said, "What a book a devil's chaplain might write on the clumsy, wasteful, blundering, low, and horridly cruel works of nature. What a book a devil's chaplain might write on the clumsy, wasteful, blundering, low, and horridly cruel works of nature." That echoes a lot of what evolutionary biologists have said. It is a half-truth. It is a half-truth. The works of nature now are cruel. It says in the book of Genesis, the ground is now going to grow thorns and thistles, and by the sweat of your brow, you'll be working until you sink right back into the dust. It has become a cruel world. That is actually true. So we have theories of intelligent design and creationism. I informally say the difference, it's creationism believes in intelligent design, but they also believe the world is cursed. So we could say intelligent design theory says some features of life and universe are intelligently designed. Creationists are okay with that. Creationism goes further and says life is intelligently designed. Life and the universe are intelligently designed, but they are also cursed. They are also cursed. That's kind of my informal definition that distinguishes intelligent design from creationism. Mm-hmm. So yes, there is waste. There are horrible works. And in fact, I should have shared my screen so people could see this. I apologize. I didn't realize my screen wasn't shared. So here's the book here, The Devil's Chaplain. And what a book a devil's chaplain might write on the clumsy, wasteful, blundering, low, and horridly cruel works of nature. You see, when Adam basically gave away humanity to the devil, um, the Bible teaches there's this God of this world. It means that there is a lot of people's hearts and minds are under the dominion of the devil. That's kind of, you know, Jesus referred to some Pharisees as the children of your father, the devil. And so there is dominion in this world that is satanic. It's really sad. I mean, the Lord is Lord over all, but he's allowed it to go to pot. And so, yes, what a book a devil's chaplain might write on the clumsy, wasteful, blundering, low, and horridly cruel works of nature. This world is horrible. But as we've shared today, there are still evidences of a phenomenal design of the original creation. God has still left he's left the testament of his wrath upon us by all these horrible, cruel things, but he's also left a testament of his genius and his ability to create things beyond all human capability that shows his wisdom and power and knowledge. And so it's been a pleasure talking through this journal club, kind of reflecting theologically, but then also touching a little bit on the science. So with that, I'm gonna encourage my co-host to rest and wish him a happy new year.
Likewise, Sal, thank you very much for having me.
And thank you. And I'd like to wish all the believers and those who are seekers a blessed and happy new year. And I want to thank you all for joining us this afternoon. Take care and God bless you.